Good morning, students. Today we will discuss regarding the principles of inheritance. We have studied it in class 10th, but once again, I would like to focus on the terminology because if we know the terms, we can easily define the techniques how the inheritance of characters run from parents to their offsprings. So there is certain terminology. As we know that this is cell, and inside the cell here is the nucleus. This is nucleus. Here are the chromosomes, right? And these chromosomes contains the DNA, the histone protein. This DNA contains so many small stitches which are functional and that are termed at the genes. So before this one, if we want to know the chromosomes, chromosomes are present in pair, right? This pair, this is termed as the homologous chromosome. Clear? Here is the another chromosome. This is the homologous chromosome. This is the another set, but this and this, these are the heterologous chromosomes. Clear? Means a set of chromosomes. As we know that we have 23 pairs or 23 set of chromosomes, right? So that set means one set, two set, three set. These all are the set means group of homologous chromosome. One set means there are two chromosomes that are the homologous chromosome. The next part that is the heterologous chromosome. Okay? And if we are considering this homologous chromosome, these chromosomes have these are the sister chromatids, right? And these chromosomes possess the genes. Suppose it is A, this is B, C, and D. And the next arm has the. I'm making a separate diagram, right? You know, gene is the stitch of genes, the stitch of DNA, which is functional or active for making any kind of proteins. This is A, B, C, right? This is A, B, C. They are the sister chromatids, and these sister chromatids have the same genes in half. It's one sister chromatid, and then the same as in the next sister chromatid. What about this? So this is very simple, but these genes may change. Certain changes may occur, but still they are performing the same characters, responsible for the same characters. Suppose this A has certain changes and now we are denoting it as A. If this B may be certain, have the certain in it, denoting the B. And likewise small c. Okay? Means these A, B, C, they have the characters. means quality of organism which makes some different than others. So this is the character. So first of all, that are the chromosomes, two types, homologous and heterologous. Okay. The next thing is the character and the third. Suppose now they are getting what it means. It means suppose it is responsible for a character. Suppose height. So a long, a small, a dot. So these are the traits. The genes present in same location of its sister chromatid is termed as alleles. Alleles like this is a single allele, this is the next allele. Okay, so these are the alleles, means capital A, small a. If there may be certain changes occurs in this A also, so it makes the next allele a nice. Again, there may be certain changes. So here three alleles, one, two, three alleles are there, right? If the next cell are having the same genes, it may become the A. So here are four alleles. One, two, three, four alleles means pair of same gene present at the same locus of the sister chromatid is termed as alleles. And trait. Each character has their contrast character. Like length, it has the height, long, and dot. So they are the traits. The contrast character of a characteristic is termed as the trait. The next one, we are seeing the genetic constitution. Means if we are seeing the genes, what genes are present there, then this is termed as genotype. And if we are considering the morphological character, suppose the color is red or white, if we are considering the morphological character, then this is termed as the phenotypic character. If we are seeing the genes present inside the organism which are responsible for a particular character, then this is termed as the genotypic character or genotypes. The next thing, if we are crossing between two contrast character, so maybe one character appear in the generation and next one become hide. So the character which is appear, that is known as the dominant character. Dominant character, right? The character which express in the generation, that is the dominant character, and the next character or the next it's a contrast character which is present but not expressed, that is termed as recessive characters. Right? So here are certain terminologies. Once again, chromosomes, well known. There are two types of the chromosome, homologous chromosome and heterologous chromosomes. The next one, characters, the quality of individuals which makes some difference with the others. That is the character. Each character has, as I told, some differences means that different characters or different factors of the same character is trait. The genes present in homologous chromosome at the same locus is termed as alleles. Alleles may be one type, two type, three types. As our blood group, we have four types of the blood group. Means we have the four alleles. That's why we have the different four blood groups. The next one is the genotype and phenotype. Phenotype and genotype. The genetic expression means the genetic constitution of an organism is termed as the genotype character, and which is appear morphologically, that character are termed as the phenotypic character. Clear? The next thing, if we have a contrast character, one of them will express and the next one will suppress. The expressed character is termed as dominant and the suppressed character termed as recessive. We have studied that Weber von Mendel is the father of genetics, and fortunately he chose the pea plant, that is the sweet pea, Pisum sativum. You knows it, right? And why I'm saying that that was his good luck. He chose the pea plant because it is easy to cultivate. It gives their flowers or seeds within 3 months. Means fast replicating. We can have approximately three generations, 3 to 4 generations in a year, right? And we can cultivate it at anywhere. And the most important, it has the 7 characters on 4 chromosomes. What are their characters? So first of all, I'm sorry. Character, the dominant character, and recessive character. Okay, the first one is seed save. Seed save means round and wrinkle. If we are seeing the seed shape, so some seeds are round shape, some one are the wrinkle shape. So round shape seed, round shape character is dominant over the wrinkled character. The next one, seed color. That is the yellow, and the recessive is the green. Means if we are crossing between these two types, this dominant character will express phenotypically. But genotypically, both the dominant character as well as the recessive character both will be present in genotype, but express the only dominant characters. The next one is the flower color. Red or violet, and this is white. Now, pod shape, if we are considering the pod shape and pod color, so pod shape may be full or constricted, full or Constricted. Full means, suppose this is the pole, right? A constricted. Right. Next, code color. Again, it is green and yellow. This is green and yellow. The last character is flower. Position, that is the axial or terminal. Axial or terminal. Axial means flowers are producing at the axial location, and terminal flowers are producing at the terminal position, right? So here are the 7 characters. One more, someone was considering the stem height like the tall and dwarf. If we are not considering sometimes the seed shape and seed color, we are considering it a single character. And if we are not, then the plant size, it may be long or dog. So here are 7 or 8 characters we are considering in Python SciTypeO, and now further we will make the cross between them, either between the dominant characters or between the recessive characters. When the crossing will occurs, crushing occurs between these all characters. But for suitability of our study, we will consider single characters or 2 characters or 3 characters likewise. So first of all, we will consider the single character, and that why it is termed as the mono-hybrid cross. Suppose we are considering the dominant character, the flowers. Flowers are red, both are dominant, and this is yes, capital R, small r. This is red flower, and this is white flower. As we are seeing that they have the same type of alleles, that's why this is known as the homozygous. And here the smaller, here are the both same, that's why it is considered as the homozygous, right? If alleles are same, then it is homozygous. If alleles are different, then it becomes the heterozygous. So these flowers make the gamete. As we know, this is the 2n number. We are seeing here 2n number of the chromosomes. When the gametes form, then only n number of chromosomes will present. That is the haploid number, right? So these are the gametes. Gametes. and this is the parental generation. Now, all the gametes are free to cross with each and every partners. Suppose this gamete will cross with that gamete. Then what will be the genotype of progeny? It has the capital R and this is the small r. Small r means this is the homozygous, but we are getting heterozygous. The next one, suppose this gamete cross with this gamete. Okay, again we are getting capital R, small r. Suppose we are getting this one, capital R, small r. This one, capital R, small r. Is that clear? Now what happens? We are getting 100% heterozygous, flowers, right? Genotypically, and if we are saying this is the offspring generation or upon generation, and all the plants are red, as we know that this is the dominant characters, so all the plants we get red. This is the F1 generation. Now what happens? When we are getting this heterozygous seeds, we are producing them once again, and when we get the flowers, again, suppose we are crossing between these two, okay, right? Then what will be the gametes of these one? Capital R, small r. Similarly, it has the capital R, small r. The rest of the process are same. This is a from generation, right? This is parental generation. Now, what happens here? As I told that any gametes can cross with another gametes. There is no restriction at all. So, suppose this crossed with this one, both are capital R. Here is the homozygous, right? Suppose it will cross with this one. Here is the heterozygous. Suppose this will cross with that gametes. This is the heterozygous, and suppose it will cross with that one, homozygous, right? Now see the color. It shows a red color. It shows a red color. It shows a red color. We are getting here also the red, and red, right? But what about this one? What about this one? Actually, this is also homozygous, but a recessive homozygous. That's why it shows a white color. What happens here? This is the F2 generation. In F1 generation, 100% flowers are red, but in F2 generation, 1, 2, 3, that is the 75% flowers are red and 25% are white. This white comes from what? Because in F4 generation, all the flowers are red. It means there are certain changes between the genes, and now they segregate in their gamete formation. That is the main important. See here, what happens? Here, the dominant and the recessive gametes both are same at the same position, and the dominant one is expressed, right? And it shows the red color. And during the gamete formation, this red is separate, and recessive red will also separate. white color, right? And they cross independently, right? So what happens here? Both segregate. So it shows 3 is to 1 ratio, as we have seen here, right? So this is the phenotypic character. This is the phenotype. We are seeing the morphological characters, right? And what about if we are seeing the genes? So this is red, okay. One, they both are red, but heterozygous. So two, and here is the white color, right? One, that is one. So this is the genotypic character. Once again, if we are considering the morphological characters, so red, 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 3 red, 1 white, so ratio is 3 is to 1. This is the phenotypic ratio, right? And if we are considering the genetic combinations, so we are getting 3 type of combination: 2 homozygous, 1 dominant, 1 recessive, and 2 new recombinations that are the heterozygous, and thus the genotypic combination or genetic ratio is 1 is to 2 is to 1. When Mendel do this cross, we are considering here single characters. That's why he termed it monohybrid cross. Because here Mendel considered a single character, right? That's why it is termed as the monohybrid cross. What is the monohybrid cross? This is also termed as a law. As here we are seeing the gametes are segregate. So this is defined, that was the postulation, and the law was termed as the law of segregation. Clear? Law of segregation. Because gametes are segregated here. Now, as we are seeing this monohybrid class, and want to know that which type of class is this, the purity of gametes, because we are not seeing the genes. So we do just back side cross. Okay? This is the F1 generation, and that one is the F2 generation, and this one is the L. Now, what about the back cross? Means if F1 generation progeny cross with the parental generation, then it is termed as the back cross. Okay? When this one cross with the uh, parental generation comes, then it is termed as the back cross, means progeny cross with the parents. Okay? So what we are getting? There will be 2 types of cross. This is the F1 generation progeny, and this is the dominant parental parent. So here the gamete formation, uh, uh, the gamete combination, capital R, capital R. What about the cross? Capital R, capital R. This is the capital R. This one, capital R, capital R. This one, capital R, small r. All are red. All plants are red. We are getting nothing. This is the back cross. One more back cross is there. This is the F1 generation and the recessive parent. Recessive parent. Yes. Now, this one is capital, small, small, and small. Next, capital R, small r, capital R, small r, small r, small r, small r, small r. Here we are seeing that 50% approx 50% red and 50% white. What it means? Here are two type of progenies, homozygous progeny and heterozygous progeny. Right? We don't know these three flowers are red, but I don't know whether it is homozygous or heterozygous. Okay, so how I came to know? Again, if I am getting crossing with that progeny with its recessive parents, so I am getting the same result, all are red. Okay, it means it is homozygous. And if this red flower plant is crossed with its recessive parent, then we are getting 50% red and 50% white. Here we are getting 100% red and here we are getting 50% red. Why it is? Because that red flower plant is heterozygous. So whether it is pure or hybrid, we are getting this conclusion that plants are pure or hybrid. So we test it. That's why this is termed as test cross. To check the genotype, whether it is the homozygous or heterozygous, we are taking the test cross. That is, there are two types of the back cross, from the dominant one and next one is the recessive one. So if the F1 or F2 generation progeny are crossed with recessive parents, then it is termed as test cross, and this test cross are doing to get whether these are the homozygous or heterozygous, pure or hybrid. Clear? So today we have discussed up to the monohybrid class, that is the law of segregation, and back cross and test cross. In next class, we will discuss regarding the dihybrid class. We will consider two characters. Thank you. Have a nice day.